Hello, everybody, and welcome to the secret history living in your aquarium. Tonight, or today, or tomorrow, whenever you hear this, I'm going to tell you a story. And this story is going to fit together in an interesting way. But what we're going to be talking about is transparent fish. Fish that are clear, fish that are translucent, that you can see through, you can see their organs. Sometimes they don't even have organs in the area you can see through, and yet they're still clear. Glass catfish, glass bloodfin tetras, there's all sorts, and we're going to look at a bunch of them also. But the story of why they're clear evolutionarily, how it helps them, and also why their clarity has shown us clarity as humans starts actually somewhere else. And it starts up in space. And with the 1980s missions to the space shuttle. So let's take a trip now and discover a little clarity. So it has been known for years that looking into translucent fish allows us to study things that we would normally never be able to see. As you can see here in this young juvenile ancestral bristle nose pleco, we can actually see its heartbeat. We can also see its intestines and the curling of its, waste matter. We can see its mouth, the formation of its teeth, and its skin and the glands, and we can actually trace the blood moving around the body as that heart beats every second. Well, this was studied from the 1920s, 1930s onward as a way of watching how human hearts and tissue and organs move and develop. And it was found that juvenile fish were a great example of a species that we could use as a human analog. As you can see here, as this fish moves, it's getting more and more energetic, and its heart is moving faster and faster and faster, and you can see that it's moving more blood and contracting and constricting over and over, releasing, and you can actually make out each valve. And so we can take animals, fish, and all sorts of clear animals that we know have similarities to humans, and we can study them and learn a whole bunch about ourselves. The problem is these are rather small, and as they get older, they lose that translucency that is so dear to us when we're studying things like cancer or heart disease or heart malfunctions that people are born with. So what did we do when we were talking about that space station? What was the solution, and why did we need to go to space? Well, the answer is it all leads back to the Danio. This fish that we see zooming around with the long fins is also an example of humans modifying a species to fit their own needs or desires, I suppose. And here we can see barbs and other fish swimming around too, but danios, and specifically these leopard danios, as well as zebra danios, or as they're called in scientific papers oftentimes, zebrafish, they are a great human analog in that they share many systems in the body that we do. In fact, their nervous system, their spinal cord, their eyes, their brain stem, a lot of that stuff develops very similarly to our own. But as you can see here, they're no longer clear. They have dark spots, they have yellow pigment in their fins, They, well, they move around a lot too. That's another tough part, but we can always fix that, right? We can always use some clove oil or something to settle them down and take a look at these functioning systems while they're alive and healthy. Right, so back for a moment to the amazing complexity and diversity of these zebrafish or leopard fish, but really it's the Danio or Dario Rerio. And these are taken up into space because they're the hardiest fish that we know of. And in the late 1980s, we figured out a way to turn their entire body clear, not just the tail fin or the, the dorsal fin and pectoral fins, not just part of the tail, but we actually were able to track down what makes fish translucent, why there are species of fish that you can downright see all the way through. And we took that information about specific genes that were used in that evolutionary process, and first we figured out the gene for the black dots or pigmentation, the melanin based pigments. Then we figured out the color based other colors you see in fish, the blues, the greens, the yellows, the oranges, the reds, every color that you see in fish. You've either got your dark melanophores, which are the melanin in the skin or scales, the flesh of the fish, that cause these black and dark patterns. Or you have the chromatophores, which are causing other colors like reds and yellows that we see in other fish's fins, or blues for that matter. And again, we come to the thought of what if you could turn that off and we could see through into these adult fish. So with a bit of research and the cutting edge genetics, which were brand new to the time, we were able to study something other than just invertebrates and crustaceans, shrimp or fruit flies and frogs and things that we had traditionally dissected. And we were able to actually dissect custom made, and not just dissect, but observe live specimens that were healthy, custom made versions of these fish. So you've got the melanophores, the chromatophores, and there was one more piece to the puzzle that's very apparent when we take a look at these fish right here, and that is the erythrophore. The erythrophore is what causes shimmer in fish. It's what causes that brilliant blue color. So the melanophores you see are causing the dark black pigment, and if we could turn that off, you just have clear fins on this little goby here, which is a Stiphodon simoni. It's a cobalt goby, this one in particular. And remember, we want those fish, the zebrafish, because they happen to share a lot of developmental similarities to humans, surprisingly, but also because they're super hardy. They can live in tanks like this one here that has no filtration. They can be in hot water, cold water. Check out my species profiles on them. And for that matter, check out my species profiles on any of these fish, on all the Stiphodon species, on the angelfish, on the coolie loaches, for more information about them. But back to the wonderful zebrafish. We are able to study these, and in their later life, they actually progress and age and have even cognitive defects that are reminiscent of those found in humans. So in the 80s, we used our genetic know-how and we began to actually turn off the genes that cause things like color and erythrophores and the melanophores, or the melanin. So you can see in these fish here, you see that sparkle, that blue color. or the yellow in the fins. Up here on these Pseudomagil luminatus, they're very beautiful fish, and they sparkle a yellow and also a blue color. Well, those sparkly colors are actually tiny crystals in their flesh, in their scales, called Erythrophores, and they're little clusters of cells that are little prisms, like the cover of that famous Pink Floyd album, Dark Side of the Moon, where you have light shining through a prism, a triangular prism of glass, and you can see all the different colors split up from the white light. Well, it works the same way in fish, and their Erythrophores are used not just because they're sparkly and colorful, and yes, they probably attract mates and attention, but they also are a defensive weapon or defensive mechanism, in that these fish can hide amongst an open area. Even if they're used to hiding and darting down into the rocks like many of these other fish do, these fish that hang out near the top are covered in Erythrophores. And if we look down at other species of rainbow fish, we can see that these Gertrudes also have Erythrophores all over. And yes, they have some melanin pigmentation, and they've got a little bit of white coloring and some yellow, but they also have that beautiful sparkle and shimmer, and it's so that predators below them will not see them because it reflects the UV light, just like that Dark Side of the Moon album cover. It splits it up into pieces and projects it all over. Another great example of that is this fish we're looking at here, which it has almost no pigment or chromatophores other than on the tail, and it's sheer Erythrophores. This is a half-beak. And by the way, all these fish happen to be from aquaticarts.com, and I've linked a code in the description if you want to get a hold of any of these amazing fish. They provide most of the fish in my fish room, and they're my favorite folks. So try them out if you haven't checked them out before. But as I was saying, these beautiful fish, they shimmer from on top, these ones. That's why oftentimes you'll see fish with bellies that are one color and bodies on top, their back is another color. And when they spawn, when they mate, those patterns can change, and it's often based on where they're living in the water level. Well, remember, so these half-beaks, these are golden wrestling half-beaks, they're living up high, and you can see that UV light is reflecting really badly off the top, so a bird or something could find them. However, they live in jungles. They live in slow-moving waterways and creeks and in very thick vegetation like this. So from below, when we try to spot them, there's very little of profile to see, and also it's hard to see them. That light is reflected off the sides of them, and they actually cast less of a shadow when we look straight down on them than they would if they didn't have those Erythrophores. So they're a really cool camouflage trick to have those Erythrophores. Now, body shape and patterns also play into this, and remember we talked about chromatophores, which are what give the color, and then the melanophores, which give patterning of darkness and shadowing, like down here. That's how fish blend in with their surroundings, such as grass, or maybe they live on rocks or in sand. And you can pick out, by looking at your fish, you can pick out what it is that they probably live on in the wild. I mean, if they have spots all over that's dark, they probably live on dark sand with light spots or with shimmering flecks in that water. If it's the other way around, then they probably live on light substrate, on sand or something. And what happens when they're translucent to that light? Well, a number of things can happen, and I think we should look at some truly translucent fish to talk about the final piece of all this puzzle. Alright, so here we have another beautiful school of fish that are missing their melanophores. So here you can see ember tetras and bloodfin tetras. that are just beautiful. And these fish, you can actually see all the organs in them. And so scientists thought, all the way back to the 1920s and 30s, surely if we could find a way to hybridize the danios and the fish that we want to study, rice fish and danios were the two in particular, surely there would be a way that they could get this clarity that they see in some fish, like the glass catfish, or these, for instance. And surely there would be a way that they could learn from adult fish and not just the juvenile fish that they wanted to study. Well, with the genetic advantages of the 1980s and early 1990s, finally that happened, and they tracked it down to 2 genes, and it happened to be literally 1 gene for the chromatophores, 1 for the melanophores, and 1 for the Erythrophores. And really in the Danio species, they just needed to turn off the melanophores and then that shimmer, the Erythrophores, because they already have silver to clear skin. It's already a clear pigment like these fish. Now these fish have chromatophores in their tail, and then they also have a little bit of Erythrophore cell on their body, these bloodfin tetras, whereas these ember tetras have chromatophores and Erythrophores, but no melanophores, just like these ones up here don't either. And if we take a look at other fish in the tank, these guppies have chromatophores and lots of Erythrophores. They're really reflective, but no melanophores either. So a group out of Boston Children's Hospital decided to track down the genes. And they figured out first by using actually guppies and rice fish that they could turn off these genes. And so they selectively tried turning off genes. And some of them were not such great outcomes. They sadly killed the fish. But remember, they needed a hardy fish to go up into space so they could study it in 0 Gs. They needed to be able to watch it for its whole life. And so they started putting together all these things we've learned in science. And the reason they wanted to study it in space was as a control for various things like the effects of gravity on the circulation system, the circulatory system, that is, and other things. Now, you can also see here that the same exact species of fish that we were talking about earlier, the Ancestris, this is the exact same line of fish that are just 6 months older. You cannot see their heart at all. They are opaque. They're lemon tetras now, or lemon plecos now. Sorry about that. Lemon Ancestris L144As is what they're known as. And we can't see their heart anymore. So we need to figure out how to turn those genes off, and that is what they did, and they did it in the danios. So as we talk about this last chapter, I figure I'd just show you some beautiful fish and plants, and we'll talk about all of this, bring it home, and we'll talk about one last thing, which is those Erythrophores. When they're on the clear fish in the wild, we weren't sure what their purpose was other than for mating. You know, it shows they're healthy. Orange colors show that they've eaten keratin or carotenoids. It also shows blue colors and purple colors show that they have anthocyanins, which are another important chemical that also show that they usually have vitamin B as well as other things in their diet. Well, the anthocyanins are very apparent in these gouramis, but when we look at this from an evolutionary standpoint, the predators and things that were hunting these clear fish that had Erythrophores were having terrible luck. Something like 50% less luck when lunging each time after a fish than their counterparts that had the chromatophores and Erythrophores. So they started studying the glassfish, and I should have links in the community tab to all of this info if you're interested in some of these studies we talked about, and info in the description down below to the links of where to find these lovely fish. But when you look at light hitting the water, we see the visible spectrum here. And it shines, it shimmers, and if you look at it against the wall here, you can actually see the bands. You see that there? The bands of different light, and they're each color diode on the water. Well, UV light is light we can't even see. And these clear fish, like these guppies up here, or like those bloodfin tetras, these clear fish, they actually refract and reflect that light all over the place. And it looks like a disco ball if you are able to see UV light like many of their predators are. And so it looks like one fish is an entire school, and an entire school just looks like a chaotic mess. And so it's a really, really effective way to get away from predators. Not only this, but they don't cast the same shadow. They cast an irregular shadow. And with their organs and things compressed up by their head, their entire back of their body can become something different. It can become, they can have an eye mark on their tail, or stripes, or some other feature like these nanochromas here. And they can blend in with those colors and things, but they're not opaque. So when they're out in open water, they're spotted. If there's light backlighting them, they're spotted. But like these pencil fish, light does shine through somewhat. And so they look like these skinny black lines of organs in high light. Well, most of these fish live in tropical locations of low light, not even as bright as this tank. And that's where all of this comes in. And you need that total clarity of the total translucency that we see in these fish. So with this total translucency that we see in these beautiful fish, they found that the genes that they needed to turn off to get rid of that final shimmer so that they could really see through and really see the organs, the blood pumping, even the nerve cells under the microscope, and study some of these diseases, was this first was the gene, the MITFA gene, and that controls the melanophores. That controlled any last dark bits of pigment on them, like the dots on this endler here. And then they turned off the erythrophore, the last shimmery genes. The chromatophore genes are actually the same and similar as what happens in albinos or leucistic fish. Now, leucistic is like albino, but they still have one or two colors, or they still have red and yellow, but their melanophores, their dark colors, are gone, and their black pigment is gone. And that's why you'll see blue-eyed fish like the ancestress peeking out here that's blue-eyed, and that is the mother of the fish we were looking at the heartbeat of earlier. And that is why. And the father's down here. He's turned this peachy orange color now that he's 3 or 4 years old. And because of that gene that controls color, they can hide better in some cases, like on blonde sandy material. But these fish have been genetically modified, and we were talking about fish in space. So back to fish in space. The Erythrophore gene happens to not have an actual name. Well, it didn't at the time. And so it was just basically denoted as the narcray or narsray allele. And so when you write it out in a Punnett square and you're trying to figure out the genetics of this, you need to have this as it's a recessive trait that they have to turn off. So they were actually able to not go in and genetically engineer the fish like we can nowadays, but in the 80s they were able to find specimens that had traits they desired, and they just needed to find a mother and a father that both had that trait, and no dominant trait of color or melanophores, erythrophores, or chromatophores. And that left us with translucent danios, which there's an article with a link to, and you can see the different stages of developing that fish and what they look like and why some have stripes and dots, and all the variations they came up with is part of why we have so many in the hobby now. But they were able to use all of this info, and together they were able to figure out several very, very crucial cures for childhood cancer, including groundbreaking work in leukemia, as well as several other groundbreaking discoveries in developmental issues, both with neurology of how nerves form in young humans and young fish, all the way from the embryo, which is translucent in most of these species of fish, which is handy, and the embryo of humans, which then we can see later on with better and better ultrasound and MRI imaging. So after all of this, they end up with a fish going to space that they actually trademarked the name of, and it's known as the Casper Danio, or the Casper fish, alluding to Casper the Friendly Ghost in pop culture. But now we've been able to do this with many species, and so you'll find all sorts of different species that are either leucistic, and that's what the lemon tetras are, or that are translucent, like these guppies that don't have the Erythrophores, the chromatophores, or the melanophores. So really interesting. And, you know, we've been able to also breed color into fins and things. And now we're going to be able, using CRISPR technology, to actually insert genetics into fish and insert, you know, the genetic code for fins growing to be clear. Now we can insert the code for them to be glow in the dark, like glowfish, or UV reactive, to be more specific. They're not truly glow in the dark. But like it or not, this is a new age in science, and a lot of it goes back to studying these little fish and their translucency, their clarity. So next time you look at a fish that's translucent, I hope it sheds just a little bit of clarity on some of the amazing things going on in the leaps from your fish tank and from nature, why we need to conserve things and why we need to save these habitats and these fish that may have rare traits that are only found in one or two species, and ways that really impact human life and inventions and discoveries that are found only because of these unique organisms. Thanks again. If you're interested in finding out more info or supporting the channel, The Secret History Living in Your Aquarium, or getting some of the fish, all of which featured have been from Aquatic Arts, kind of by dumb luck tonight, check out the links below. And again, for members of the channel for $1.99, you can get the links to the scientific articles that went into the research here. Thank you so much for your time, and if I've earned it, I'd love a little thumbs up and subscribe, or maybe even a share, but don't feel obligated. Just have a great night, treat your fish well, your plants great, and of course take care of yourself so that you can keep up with that routine. Take care, guys. I'll talk to you next time. Bye.